Okay, I'm Roger Tsien, and today I'm going to talk to you about fluorescent protein indicators. In other words, molecules that have been engineered out of using fluorescent proteins to then report to us crucial biochemical signals, especially their dynamics and spatial localization inside cells. So basically there are four major classifications of how these indicators have been made. And they're listed, first two are listed here. The first and simplest class in some ways are the translocating fusions in which, in order to monitor the activity of enzymes such as PI3 kinase, that's phosphatidylinositol 3 kinase, or protein kinase C, calmodulin kinase type 2, etc., G protein coupled receptor desensitization. In all of these cases, the signals have caused a binding protein to move around inside the cell from one location to another. And all we have to do is tag that binding protein or motif with a fluorescent protein and watch where it goes. And when it goes, if we did our biochemistry right, that'll tell us that the original signal was generated. And I'll give you one example of that. The next class is when we engineer a GFP or YFP to monitor things like directly pH or indirectly chloride, redox potential, calcium, and so on. And all of these are by introducing mutations into the GFP and sometimes whole chunks of other proteins that enable the equilibrium between the tyrosine anion and the tyrosine neutral forms of the chromophore to be altered by the biochemical signal we're trying to measure. And all of these just use a single fluorescent protein. Then a large class of indicators is based on pairs of fluorescent proteins, typically of two colors, where they are the shorter wavelength one can do fluorescence resonance energy transfer. And it serves as the donor. We excite it directly, and if the other protein of the longer wavelength, which is called the acceptor, is within a few nanometers of distance and is appropriately oriented, then the longer wavelength protein steals the energy from the shorter wavelength protein and re-emits, typically. But in any case, the efficiency of that energy stealing or energy transfer depends on distance and orientation. And therefore, any biochemical signal that changes the relative distance or orientation between our pair of fluorescent proteins will cause a change in FRET, F-R-E-T. And in a way, this is FRET, I should remind you, is somewhat similar to the basic way that the GFP in the jellyfish took the energy from equorin. Equorin glows blue. In that case, it's not fluorescence, it's bioluminescence, but once you make the excited state, it doesn't matter. And what the jellyfish modulated, or to change blue to green, is what we deliberately modulate in order to measure all sorts of signals ranging from calcium, cyclic AMP nucleotides, protein kinase activation, the activation of proteases, even membrane potential, and all sorts of protein-protein pair interactions. And finally, I'll give you one brief example of bimolecular fluorescence complementation, which uses the special property that some fluorescent proteins can be cut into separate halves. Not literally that we started as one complete half and were cut by a protease, but rather we synthesize them as two separate fragments, and those fragments don't work and don't become fluorescent by themselves, but if they're welded together in proximity for a little while, they can find a partner and generate a fluorescent protein out of their two separate halves. Therefore, anything that brings them into proximity, these fragments, will generate a fluorescence signal. So here's the translocating case. This is class number 1. And I only have this one example, but it's pretty simple, in which the activation of phosphatidylinositol 3 kinase, in this case triggered by the addition of platelet-derived growth factor to these fibroblasts, these NIH3T3 cells, will generate the unusual phospholipid phosphatidylinositol 3,4,5 trisphosphate that's called PI345P3. And we have a domain that's long been known. In this particular case, it was stolen from a protein kinase called AKT, and the domain is called the plextrin homology domain. That's why it's called pH. And that piece from AKT was fused to GFP and expressed in the cytosol of the cells. Now, before we stimulate it, this GFP is just sitting in the cytosol like, diffuse GFP, and there's nothing much to see. But that's just before we added the PDGF. When we add the PDGF, or when these workers added the PDGF, this is from Tobias Meyer's lab, in the next few minutes, that growth factor acting through the tyrosine kinases initiates a signal transduction cascade that generates this lipid in the plasma membrane, and then the AKT pH domain notices that there's this lipid and wants to go bind to it. So when you look carefully at this micrograph, you will see that the GFP is not completely, but to a large extent, jumped over to the plasma membrane and lit up the plasma membrane and left the inside of the cell a little bit denuded of protein, somewhat denuded. And then later on, just to prove that this was reversible, they put on a drug that inhibits the kinase, and that's called wart-mannan, and that allows the PI345P3 to be broken down by other processes, and the whole process reverses, and the GFP goes back to being in the cytosol. And this is the time graph of the relative intensity of the plasma membrane compared to the rest of the cell, going first up with a little bit of a delay due to the PDGF, and then coming back down with wart-mannan. Now let me already move on to class 2. These are the GFPs that are intrinsically changing their fluorescence properties in response to an external biochemical signal. The original GFP was already somewhat pH sensitive, and these are spectra taken at different pHs and showing that the spectrum a little bit goes up and down. But this can be improved or amplified by mutagenesis if this is what you want. And these proteins called fluorins, which is a clever pun, pH for the pH, and fluorin sounds like fluorescent. These proteins will do all sorts of things in which, for example, we can actually change the balance between the left hand or UV peak versus the right hand, which is the visible peak, by changing the the pH, or we can just make a much bigger change in total intensity, the so-called ecliptic type of fluorin. That's just a different set of mutations. And the place that this has become really widely used in cell and neurobiology is to monitor exocytosis, because when you put this protein into a secretory vesicle, you should know that the inside of a secretory vesicle is usually quite acidic when it's sitting inside the cell, like typically pH 5 or so. But if those contents get exocytosed, then the protein now suddenly sees the extracellular normal pH, which is typically 7.4. So that protein reports that that secretory granule or synaptic vesicle just got exocytosed. And we can even use this now, and has been used as in neurobiology, where we want to monitor synaptic release, and we can do so, and this is in case in flies, where different odorants were put into the antenna of the Drosophila, and now we're imaging with these pH-sensitive indicators, which are being expressed in the processing machinery, and wherever the right odor causes the cell to exocytose and do its synaptic transmission, we get a little burst of higher pH as seen by this synaptofluorin. In the case of the YFPs, it became possible to find mutants in which the pH dependence was shifted to becoming a chloride dependence. Well, not entirely shifted, it's still pH sensitive, but now at a fixed pH, the chloride will shift the effective pKa of the equilibrium, and so as long as the pH doesn't change, we can also measure chloride, and this was, for example, used to screen for drugs that would modulate chloride fluxes. And the place that this was clinically important is that people who suffer from the the disease called cystic fibrosis have a problem, a mutation in their protein called the CFTR that conducts chloride and bicarbonate, and it was then possible by putting the YFP into cells and exposing them to different chloride concentrations and test cases to monitor whether or not the cells were responding to chloride that was being changed outside, or in this case iodide, and see whether the fluorescence was changed, and determine whether or not drugs that could be applied were fixing the function of CFTR. Now, for the case of calcium, which is one of the most important messengers in lots of signal transduction, including neurobiology, it turned out to be necessary to modify the GFP even more drastically. Original GFP has an N-terminus and a C-terminus like any old protein, and the sequence at the N-terminus happens to start with MV, S, K, etc., and ends at 238 amino acids later with this particular lysine. But there's been a trick known for a while that sometimes works on many proteins, not always on every protein, which is that if you can recode this protein so that you have basically the same amino acids, except that you start them in a new location and end them in a new location, and in the process you also have to link the old N-terminus and C-terminus together with a few amino acids. This, by the way, does require that the N and C termini of the protein you care about be fairly close to each other in space so that you don't have to make a very long linker. Sometimes this so-called circularly permuted protein will actually get up and work. And in this case, what it has to do is become fluorescent. So you can express this modified protein, which starts in the middle of what used to be the middle of the old protein, go around, and this will become fluorescent. So in three-dimensional terms, this was the 11-stranded beta barrel of GFP with the alpha helix running up the center and the chromophore in the middle, and the N and C termini were up here. That was the old N-terminus, and this is the old C-terminus, and instead we link these two together genetically and make a nucleic acid that encodes a few amino acids to connect them. But we start the protein now with a new methionine here. That becomes the new N-terminus, and it runs through this the set of strands around the barrel, more or less as the old one did, except it starts from a new place and ends in a new place. And these proteins become fluorescent, and they're a little bit more fragile than the original GFP, and that turns out to be useful because we can then take this old new N-terminus and fuse onto it calmodulin, which is a famous calcium binding protein, and that actually went onto the C-terminus of the so-called circularly permuted enhanced GFP, and then onto the into the new amino terminus we fuse a peptide called M13 that calmodulin likes to bind to when it sees calcium. So basically we've sort of created a wound in the side of the fluorescent protein, and then that wound sort of hangs open and depresses the fluorescence until the calmodulin sees calcium. Then the calmodulin grabs onto the M13, they lock together and they sort of locally stitch up the wound, and we restore the fluorescence. And this can all happen in just milliseconds or so, or a few hundred milliseconds, which is the speed of calmodulin. And this has become very popular. It's called GCAMP, which is a sort of a pun on calmodulin being stuck inside GFP. And there's another flavor called pericams, and these have been widely used now, especially in neurobiology, to follow calcium transients, which are again good markers of activity. In this particular case, the people doing this work are studying the activity of parallel fibers in the cerebellum, which are being electrically stimulated. And when they are electrically stimulated, they let in some calcium through voltage-operated calcium channels. The GCAMP is expressed inside these cells and momentarily flashes bright. And with that, we can get spatial maps, again, of where the neural activity is occurring inside the cell. There's a lot more recent work, but I picked this example, which is a little bit older because it's a very simple case to explain. So now let me move on to the third class of indicators, which is not just a single fluorescent protein, but to remind you, we have two colors of fluorescent proteins. By far the most common historically, not necessarily the ones we will use for the rest of scientific progress, but historically we have tended to use cyan and yellow. They are a good pair that talk to each other nicely and can be excited separately and detected. We excite the cyan with violet light. It tries to glow cyan, but if the yellow protein is nearby, the yellow steals the energy without any photon ever actually coming out, and then the yellow then goes on to re-emit yellow-green as if it had directly been excited in the first place. And the very simplest case is you just tie the two together by a short leash. It can be up to 20 or 30 amino acids long, and then as long as that's intact, this energy transfer is very effective. But then if we come along with a protease that cuts this linker and splits the two apart, now the cyan and yellow drift apart. They're no longer held together, and all they have to get is a few nanometers apart, and now the cyan is on its own. You excite it with violet light, and it emits cyan, as implied by its name. By the way, crucially important in this is the fact that the fluorescent proteins themselves are not very easily proteolyzed. And fortunately, they are well-folded proteins, and well-folded proteins don't have loose floppy bits, whereas the linker we make is just unstructured amino acids, and typically it is vulnerable to proteolysis. This is an irreversible case. Once you've broken this thing, you cannot put Humpty Dumpty back together, and the cyan and yellow have no reason ever to get back to each other. So this is a one-shot indication that a protease was active and made the transition. A reversible case, again, uses the calmodulin and M13 motif. And remember, when calmodulin sees calcium, it then grabs onto M13. So in this case, if we then sandwich that pair between cyan and yellow fluorescent proteins, the addition of calcium actually brings the cyan and yellow closer to each other. They were relatively far apart before. They become closer and better packed, oriented with respect to each other. So in this case, calcium increases the fret, and when we take the calcium away, it can go right back to its original state. So this process can go back and forth many, many times as needed to signal. In this case, we see calcium not as so much as a single intensity increase as it was in the GCAMPs, but rather a color change, the calcium giving us more yellow and less cyan out. So two more examples, and then I'll show you some of the biology. This is a case where we can look at cyclic AMP. Cyclic AMP's original native biosensor inside most mammalian cells is called cyclic AMP-dependent protein kinase, and cyclic AMP binding to the regulatory subunit, there are two of them in this tetramer, causes a conformational change that makes the regulatory subunit spit out the catalytic subunit and stop binding to the catalytic subunit. So it's fairly obvious, hey, put one color, in this case cyan, on the regulatory, put another color on the catalytic. In the original form, they are doing fret to each other. They're held close to each other. When the cyclic AMP comes on, it breaks up the fret. And this process is also reversible. When the cyclic AMP is destroyed, then these subunits find each other and go back to this state. How can we see not just cyclic AMP, but the action of any protein kinase? As you know, protein kinases are amongst the most ubiquitous and universal and important of all sort of signaling mechanisms. And here we exploit the fact that there are certain known motifs that like to bind phosphorylated amino acids, but won't bind the original amino acid before it got phosphorylated. So now this is a variant of the calcium sensor. Now the pink bit is the floppy peptide that's in the driver's seat. It's not M13 anymore. It's a substrate that the kinase wants to phosphorylate if the kinase is active. Once it gets phosphorylated, now there's a phosphate stuck on a serine, threonine, or tyrosine, and if we've taken this binding domain correctly, it will then want to eat the phosphate or grab onto the phosphate like a fist closing around it, and that's shown here where the orange dot symbolizes the phosphate, which is being grabbed by the catcher's mitt, which is the binding domain. And in this process, we change the relative orientation or distance between the cyan and yellow. As I drew it here, it increases fret, but depending on the details of what this is really here, sometimes there's a decrease in fret, but we don't really care as long as we get a big signal. So here's some biological examples. This is the protease sensor for apoptosis, and you know that in apoptosis there's the massive activation of a whole sequence of proteases, including the famous caspases, and they like to cut the sequence DEVD. That's what, their favorite substrate, known from other biochemistry. This isn't all there is, but basically this is the crucial bit of the substrate, and we put that between CFP and YFP. We get strong fret beforehand, and in this particular case, cells were induced to apoptose. And what's interesting is that in any one cell, the transition is very sharp. And it only takes a few minutes to go from complete fret to complete destruction of fret and complete proteolysis of this molecule. And the reason that this is biologically important is that up to now, before these experiments, people mostly had to watch apoptosis as a population of millions of cells by the traditional biochemical techniques. And when you look at millions of cells, they each do their transition at a different time. So in the population, the transition between not active and active caspase takes hours. But when you can watch it in single cells, you see that every cell does it in a few minutes, but that different cells do it at different times, and that's what generates the spread of times. A different form of proteolysis that doesn't actually use fret, but very beautiful and interesting example is shown here, where the Milwaukee lab decided to make an indicator for the cell cycle. And in the cell cycle, we also have sets of proteases that turn on at different phases. And particularly, we have some proteases that destroy proteins during mitosis, and other proteases that destroy proteins every time but when you're in mitosis. And they managed to put YFP under the control of one of them so that YFP is it is highly expressed during mitosis and destroyed at other times, whereas the red protein, M-cherry, is exactly opposite, and it's made during and persists when the cell is not in mitosis, and then during mitosis it gets destroyed. So they're exactly out of phase with each other. And the fortunate, easy way to remember this, these workers picked the color scheme right so that green means go, means you're in mitosis, and red means stop, like traffic lights. They could perfectly well have done it the other way around, but it would be harder for us to remember. And so here's the movie of a time lapse of 1 week's worth in the life of these two cells, which are dividing. And as they divide, they flash from green to red, to green to red, to green to red. And we can watch the cell cycle biochemically, and all this, the highly controlled activation and destruction of these proteases. Now you might say, why did you need this? Hey, we could have watched it, just by seeing when one cell split into two. We knew when mitosis occurred. So why do you need a biochemical indicator? Well, the place you need it is, say, when you're going to work in an intact mouse, and now you have a cancer that's been implanted into the mouse, and you don't have the spatial resolution nor the continuous imaging to watch every single cell to track when it does mitosis and follow the history of every single cell. But now, just by looking, we can see that this cell population, 1 day after implantation, is a sort of yellowish color, which is a mixture, therefore, of mitotic and non-mitotic cells, but by 16 days later, it's all red, which says that even without high-resolution imaging, all of these cells have stopped dividing. That's what makes this a benign cancer cell. This is a malignant cancer, and even through the flank of the animal, it is visible. You can detect that it's continually dividing. And when we then do microscopy, we can see the distribution of the mitotic cells, which in this case are close to the blood vessel, and the non-mitotic cells that have no longer rapidly dividing, which are further from the blood vessel. And this is just from a single snapshot without having to watch the entire movie for 27 days. So here again are the chameleons, which is a blow-up, showing again the mechanism by which calcium can control the fret between a cyan and yellow fluorescent protein in a reversible way. And just to show you a little more about, in this case, the technical detail, we monitor the emission in the yellow band from the YFP as well as the cyan band from that one. And what calcium does is it takes away cyan and gives it to the yellow band because that's the increase in Fred. And this ratio between yellow and cyan is particularly helpful to cancel out all sorts of artifacts like the absolute amount of protein, or the absolute strength of illumination, or the absolute sensitivity of your camera, or all these sorts of fluctuations are canceled out when we take the ratio of two wavelengths. And so here's an example from the earliest form of development. This is a zebrafish embryo that has just been fertilized. It is expressing this chameleon, as we call it, which is again a pun on the word calmodulin and the fact that it changes color. Here, this is 30 minutes post-fertilization, and as this zebrafish begins to divide, you will see cleavage furrows form, and each time a cleavage furrow is beginning to form, there's this flood of high calcium preceding the actual constriction, that belt tightening that then makes the next cleavage furrow. So that's 2 cell divisions into a group of 4. Now we're going into 8, and that's the next cleavage furrow. Time going by in minutes in time lapse. And so this is demonstrating the tremendous importance of calcium in causing the motility and guiding development in order to decide where the cleavage furrow is going to occur. As another example of the biological usefulness of the chameleons, and particularly here it was useful to have a ratiometric indicator that changed color, which made it possible to try to calibrate the actual yellow to cyan ratio in terms of an absolute calcium level. And this was work on the effects of Alzheimer's protein, the famous A beta being misregulated and expressed. Now first we have here looking at a normal brain. This is actual in vivo microscopy in the intact brain. It's lightly labeled. You don't see all of the different dendrites and axons because the genetics only cause maybe 1 in 100 or 1 in 1,000 cells to express, but we can see these sort of like tree trunks and roots of the different neurons, and they're all pretty much a uniform blue-green pseudo-color. This is not a real color. This is the computer coding of what the ratio is, and this is the color scale, and a blue-green is somewhere around here, and that tells you that the average calcium was on the order of about 80 nanomolar, and it was quite tightly distributed in this histogram. This is the normal brain, but after you have made the brain express the Alzheimer's protein with the appropriate mutations, then it turns out that some, not all of the processes start to turn this, have a higher ratio of more Fred, indicating more calcium. And there's still a normal population, which is more or less the same, but there's this additional small population, which actually turns out to be about 20% of the processes that have this elevated calcium. And this lasts sort of is continuous. It's not a sudden, minute by minute fluctuation. This is high, and it turns out that these are highest when they are close to the plaques that are the damaging deposits made of these mutants. So here was essential to be able to calibrate the calcium scale here in absolute terms, and that's much more difficult with all of the other forms of indicators than this 2-color version. A quick example on the cyclic AMP. Again, this is the basic mechanism that I've already described, where the regulatory subunit binds cyclic AMP, splits itself off from the catalytic subunit, thereby disrupting the close localization of cyan with yellow. Here, that pair of constructs, remember, these are 2 separate proteins. There's the regulatory that has the cyan and the catalytic that has the yellow. They both have to be transfected at roughly equal amounts, in this case into a cardiac myocyte. And this was the first evidence that really demonstrated directly by imaging that the cyclic AMP elevation caused by different agents, such as norepinephrine, which is the hormone inside you that when you're getting excited and your heart is beating faster, it speeds up the heartbeat, but it doesn't actually raise the cyclic AMP uniformly. It produces micron scale variations within the heart muscle, and that was shown here by this imaging of the activation of this indicator. And that turns out to be very important for the biology of agents like these beta-adrenergic agents, that they have a localized cyclic AMP signal, not just a general widespread one. And this is the kinase phosphatase reporter, again using the substrate that gets phosphorylated, and then that substrate, if phosphorylated, then binds intramolecularly to the phosphoserine, phosphothreonine, or phosphotyrosine binding site, and the CFP and YFP that are attached on either end of this internal construct then change their fret, if we have arranged the engineering correctly. And this movie is sort of cute. This is a fibroblast that is expressing the a reporter for the activity of the epidermal growth factor receptor. That's a famous tyrosine kinase. It's when epidermal growth factor binds to the outside, then it triggers the dimerization of this receptor, and then tyrosine phosphorylation occurs, and we arranged this reporter to be phosphorylated along with many other things, and then that phosphorylation causes the internal binding of an SH2 domain, which is a famous domain that likes phosphotyrosine, and we get an increase in fret. So here's the cell before it's been stimulated, and then in a moment I'm going to start it, and when we apply the EGF now, and there's a flood of high fret that starts from the end of this thinnest dendrite, goes up into the center, and by now the signal has largely gone away. But you may have seen that there was a flood of red that went up into the center, and then what I didn't have time to tell you is that we then washed out the EGF, and the signal went back, and the red tide sort of receded back the way it had come. And though up to now I've been talking about sort of traditional biochemical signals that are mediated, that are soluble things like calcium or cyclic AMP or so on. If you just want to watch when a pair of proteins gets together as a pair of proteins, this form of induced protein-protein interaction, it's simple. You put CFP on one end, on one of the proteins, YFP on the other. There's not a guarantee that every such construct will put the cyan and yellow close enough for good fret, but there's a reasonable chance, and you try things until you find ones that work. And in this particular case, our colleagues asked us to help them look at the binding of nuclear hormone receptors, basically steroid receptor types, with other key proteins that are called coactivators that are famous for helping drive transcription once the hormone binds. So here's a cartoon in which this is the hormone receptor, the ligand is the hormone, it comes in and causes a change in conformation that makes the coactivator bind, and so we tagged them with CFP and YFP and looked them in cells, and that's the CFP on the retinoic acid receptor and YFP attached to the so-called SRC1. It's one of the famous coactivators. We put on the hormone at this vertical line, or the drug that simulates the retinoic acid, and within 20 or 30 seconds already we have a big increase in fret that levels off rapidly. And you can do it also with the real natural ligand, transretinoic acid. Now, who cares about this? Once again, we get the kinetics. This is done in a single cell, and this showed the whole process of this biochemistry happened in just a few tens of seconds. Until this point, all the biochemical assays required that you check for the affinity of one protein for another. One of them was hung up on an affinity column, and you poured the other protein past it, and the assay took 20 or 30 minutes, and everyone thought that this sort of event took tens of minutes, but it actually takes tens of seconds, and that's at room temperature. It's probably 5 seconds or so at body temperature. My last example is now class 4, which I haven't talked about up to now, and that's the so-called bimolecular fluorescence complementation, where we take these two fragments of fluorescent proteins that each by themselves is impotent, unable to make fluorescence. And we clamp them together for a little while in proximity, and in a sort of strange variation on circular permutation, these two fragments can now cooperate with each other. In the circular permutation, they were held together, but just out of the normal order, sort of back to front. Here, we take some other scaffolding and hold them together. And if we do it right, we can generate fluorescence where there was none before, and there wouldn't be any if we didn't bring those two fragments into proximity. And this very clever application called GRASP, this is for looking at when a pre- and postsynaptic neuron get together close enough to form a synapse. And the way you do it is that you express the one half of the protein, the first fragment on the presynaptic cell and the complementary fragment on the postsynaptic cell. And if they're not close enough to quite reach across the gap, you get no fluorescence. But when they finally do get to reach across the gap, then you generate fluorescence. And down here is a micrograph of some examples where you have pre- and postsynaptic cells. Now these, just for visibility, were separately marked with other colors, like blue for the pre, I think, and red for the post. But where they come into contact, that's where the GFP, the green, got reconstituted from the two fragments. And that marks a synapse that got close enough to actually be functional as a neuron, as a synapse, as well as generate the fluorescence. So what are genetically targetable sensors good for? And in general, we now can look at some of the most important and popular signals inside cells, ranging from just pH to calcium, even zinc if you want to. I didn't talk about it, cyclic nucleotides. I didn't show redox potential, but there are other ways in which redox potential will modulate these types of signals, and membrane potential, proteases, kinases, phosphatases, and so on. Basically, just about any biochemical change that alters protein expression, localization, as in class 1, conformation, proximity, especially in class 4, any of these can be sensed if you're willing to go and engineer the molecule. And once you've got it engineered, it may take you some trial and error. Sometimes a little modeling, computer modeling has been known to help. Once you make it, then you can transfer it over and over again to new systems and look at your favorite signal that way. And the fact that it's genetically encoded greatly simplifies delivery into almost any organism that you can transfect. Or into a tissue. You can force it to go to subcellular locations, but suppose you want to see calcium in the mitochondria, not everywhere else. Well, you just put a mitochondrial targeting signal on it. Or if you want to see protein kinase activity right at the plasma membrane and not in the Golgi, or if you want to see it in the Golgi and not in the plasma membrane or in the nucleus, all of those are now controllable by the standard trafficking signals that we know about. And it makes it a mutagenic improvement. There's all sorts of tricks for doing mutagenesis and screening huge libraries rapidly for your favorite new response. And it even makes it easier to distribute around the world because we don't have to dispense fragile organic chemicals. We just have to dispense DNA, which you can just do by spotting on a piece of filter paper and dropping it in the mail, and it will survive mailing for some considerable distance. What are the exceptions? As I mentioned, at least the current fluorescent proteins that based on the jellyfish and corals cannot be done in obligate anaerobes because they all need oxygen at some stage in their life cycle to become fluorescent. And not to mention that the last one is that intact human beings do have the problem that ethically, are we allowed to transfect, say, a sick person? Assuming that we have obeyed the appropriate animal welfare regulations, we are allowed to do that into mice. We can make transgenic mice, transgenic flies, or transfect them acutely. There's nothing says that human beings are biologically different. You can certainly make these work in human cells and tissue culture, but the question of whether you can do it in an intact human being when they come to you, say, as a sick person, that's a whole other issue, which much more has to do with ethics and regulation than with molecular biology. I've thrown these 4 different sensing mechanisms at you: translocation, single fluorescence intensity, dual fluorescence fret, dual FP, fluorescent protein FRET, and bimolecular fluorescence complementation. And without going through all of this, each one of them has its own set of advantages and disadvantages. Otherwise, we could throw one of them out if it was decisively bad in all sorts of ways. For example, the translocation one is relatively easy to engineer if you've got a good binding domain already. The fluorescent protein is just along for the ride. It doesn't have to change its properties or do anything subtle. You have to be able to image it. So it's relatively easy to engineer. On the other hand, it's not particularly easy to calibrate, which is another factor. The single fluorescent protein intensity is nicely reversible very fast. Takes quite a bit of engineering. It's a little tricky to build all of this into a single fluorescent protein. The FRET indicators are the most widely used because so many things will change the relative proximity or orientation of a pair of proteins. They take a fair amount of engineering as well. And both the fluorescent single and double are highly reversible. The bimolecular fluorescence complementation, its great advantage is that it has a tremendous signal over background, because if you don't get combination, there is no signal. There's just zero. And then when you do manage to make even some fluorescent protein reconstitute, you get a big enhancement over zero. And so it gives this huge signal to background contrast. Whereas the translocation, you saw that there was some stuff already hanging around in the cytosol. It wasn't an all or nothing transition, and discriminating the two perfectly by microscopy is not always easy, and so on. In the case of the FRET, often there is barely any change in the FRET, and we only have a few % change in the ratio of, say, yellow to cyan, and it takes good instrumentation. So that has the least signal to background, but it's the most reversible. The bimolecular fluorescence complementation, once that protein has folded out of the two fragments, it doesn't need any more scaffolding. It will stay together until it's destroyed. So it's not reversible, basically. And it also takes the time of protein folding to respond. So that's why it's relatively low in reversibility and speed of response. So that's why we need all these different mechanisms, because for different applications, each one may be advantageous or not. So with that, I thank you very much, and hope that you've learned something from this lecture.